Welcome to Life in Biology. I'm Dr. Joel Graff, and this is the third video where we're working through some Punnett squares showing crosses, Mendelian genetics, and variations of Mendelian genetics. The first video did a dominant trait, which is what Gregor Mendel studied in his pea plants. The second example was an incomplete dominant trait. And then this video is going to be about codominance. And to throw in an extra variable, we're going to be dealing with a trait that has multiple alleles. Previously, we had just a capital D, lowercase d for the flower color in the pea plants. Or P, excuse me, for purple. And then with the snapdragon flowers, we had C with a superscript R, C with a superscript W for red and white for that incomplete dominant trait. But here we're going to, with the, again, another classic example of a trait that shows codominance, and that is the ABO blood group. With the ABO blood grouping alleles, you have an allele that codes for A, which is a capital I superscript A. You have an allele that codes for B, which is capital I superscript B. And then those, then if you have O, it would be a lowercase I. Now, A and B are codominant, meaning that if they are, if you have an organism or a human that has is heterozygous and has these two alleles in their genome, then they would be type AB blood. So that's what codominance means, is that both A and B would be seen as the trait. The A and the B are dominant to the final one, I, and I put sort of because it's not exactly what is meant by dominance, because this I doesn't really make anything. And so if you have the capital A or capital I to the superscript A with a lowercase I, you end up with type A blood, because this is going to be expressed and this doesn't do a thing for you. And so you have type A. Anyway, one final variation on that is that if you have both lowercase I, then that's blood type O. All right. So what we're going to do is we're just going to run through two examples of crosses for a blood group. And so in this first example, we have a cross here, and the first parent has the genotype capital I superscript A and lowercase I. The second parent has the same thing, except for rather than the A allele, it's the B allele, but we have the lowercase I. So go ahead and make your Punnett square. I'm going to put these two possible alleles. Those can be what the gametes carry, either a capital I uppercase A or a lowercase I. This organism here can make gametes of either capital I uppercase B or lowercase I. So this gamete and this gamete and this gamete come together, and you have an organism that's A and B. This one is B with the superscript I. This one is A with the not superscript, but lowercase I, B lowercase I, and then this lowercase I could match up with this lowercase I, and you end up with two homozygous for lowercase I. So the genotypes that we're dealing with here are type A, or capital I to the A, capital I to the B, and there's one box like that. There's one box that has this genotype of I to the A and lowercase I, B lowercase I, and then two lowercase i's. There's one of each of those. Each one of those squares in our Punnett square is different. Now, the phenotypes, I've written these up in the same order as shown here, is also 1 to 1 to 1 to 1, but the phenotype is what you would say the blood group is. So the character is your blood type, and the traits this one is type AB blood, type A blood, type B blood, and type O blood, and that matches up with the AB, the A, the B, and the O here. Hopefully that was clear. But anyway, 1 to 1 to 1 to 1 for your genotypes, 1 to 1 to 1 to 1 for your phenotypes. So then let's mix it up a little bit, and we'll have a cross between a parent that has type AB blood, the A allele and the B allele, crossed with a parent that has the type A allele with the lowercase. And so this parent is type AB. This parent has blood type A because that I doesn't really mean anything. Okay, make your Punnett square. You got I to the A, I to the B, I to the A, and then lowercase I. Mix and match, fill out your Punnett square, and you end up with genotypes that, again, is a 1 to 1 to 1 to 1 ratio. Each one of these squares in our Punnett square was different. However, when we look at the phenotype, type A blood happens twice, because you can have type A blood if you have homozygous for the I to the capital A allele. You can also be type A blood if you have the I to the A followed by a lowercase I. And so this first one and the third one are both type A blood. Now you've got type B blood here, and that came from this lower right quadrant, which the genotype was I to the B, lowercase I. Capital I to the B, lowercase I, that's type B blood. Now it's that person's genotype is considered heterozygous because it's got two different alleles for its blood group. And then finally, we have type AB blood was shown here, and on the Punnett square it was shown here, and so there was one of those. So we had two type A, one type B, and one type AB. Now, were there any type O bloods? None of these genotypes is lowercase I. That's homozygous for the type O allele, and so we have zero chance of having offspring from these parents that are type O blood. All right, so again, that brought up two important points. That brought up genes where there are multiple alleles in the population, and then it also brought up the idea of codominance, because A and B antigen can be expressed on red blood cells at the same time, and so then that gives you type AB blood. Hope this was helpful. Talk to you later. Like and subscribe or not.